I'm Gbola Amusa, and it's my pleasure to introduce our next guest from Tenaya Therapeutics. We have Faraz Ali, who's Chief Executive Officer, and we have Kee-Hong Kim, who's SVP of Manufacturing. The format for this session is a roughly 40-minute presentation followed by a fireside chat. If attendees have any questions during the session, feel free to type them into the question at the bottom, and we'll try to ask them on your behalf. Faraz, Kee-Hong, thanks for joining us virtually today.
Hi, Gbola, and thank you for the opportunity to present the Tenaya story at this great summit that you guys have been putting on for a few years. So looking forward to telling the audience more about Tenaya Therapeutics.
Great. Please.
For those of you who, yeah, go ahead. So for those of you who aren't familiar with us, Tenaya Therapeutics has a vision to transform and extend the lives of people fighting heart disease. And our mission is to discover, develop, and deliver curative therapies that address the underlying causes of heart disease. And with the capabilities and the therapies we're developing, we hope to deliver, hope and new therapeutic options to millions of individuals affected by heart disease, including rare disorders as well as very prevalent forms of heart disease. So some things that make Tenaya unique in this overview, Tenaya is 100% focused on heart disease. This is what we do, and we're getting good at it. Some things that are important to know about us: we're about 75 people, all based in South San Francisco. We are, some things make us unique. One is that we're advancing a pipeline that has both orphan and prevalent forms of heart disease featured in it, but we're also doing modality agnostic drug discovery. So a lot of what we do is gene therapy, but that's not all we do. And indeed, we have small molecules in our portfolio today, which we'll talk more about. Very strong scientific foundations, with both know-how and IP license from both our founders, Deepak Srivastava at the Gladstone Institute, as well as Eric Olson at UT Southwestern. We'll talk about 3 distinct product platforms that support our innovation. We have taken the decision to internalize and integrate capabilities that span all the drug discovery that we're doing: disease models, capsid engineering, promoter engineering, manufacturing. You'll hear more about obviously manufacturing today. And we are, in IND enabling studies for a lead program. We announced with our Series C, 106 million Series C, earlier this year, that we're establishing a CGMP facility right here in the San Francisco Bay Area that will be operational in the second half of this year. And we have the potential for multiple assets to be in the clinic in 2022. And now we're looking forward to telling you a little bit more about us. I'm going to skip the faces, but we've got some great people in the CSO and the Chief Medical Officer and other chairs. Today you have myself, Faraz Ali, the CEO of the company, as well as Kee-Hong Kim, the Senior Vice President of Manufacturing and Technical Operations. Something that makes Tenaya unique is the fact that we've turned the script a little bit on drug development, whereas most companies have a single platform and they apply that to multiple therapeutic areas. What we're doing at Tenaya is different. We actually have 3 distinct platforms, and all of them focus on heart disease. And we often get the question, why do you have 3 different platforms? What are you trying to do? It almost seems like you're 3 companies in one, and that is partially true. So our 3 platforms, as we tell it, are trying to solve 3 distinct problems that have plagued the field of drug development for heart disease. So one platform is the gene therapy platform, and the underlying problem we're trying to solve here are heart cells that are defective generally because of a mutation. And the solution in our hands is to use viral vectors to deliver a healthy copy of the defective gene, or to edit it, or to express a therapeutic protein. So this is where gene therapy has been for the brain and the liver and the eye and the muscle, but not as much for the heart. And that's something that we're leading right now. The other platform, we call it a cellular regeneration platform. And the problem we're trying to solve here is the loss of heart cells. So it's not that you have cells that are defective because of mutation, but the cells are gone. And they could be gone because of a heart attack or some other ischemic event. It could be a viral infection like COVID. It could be chemotherapy. The heart is, the cardiomyocytes are postmitotic. They don't have the ability to regenerate. And so our founders had not 1, but 2 different approaches for in vivo regeneration of new cardiomyocytes, again, using viral vectors. This is very innovative and very novel stuff. And then finally, there's the precision medicine platform. And the problem we're trying to solve here is discovering, identifying, validating new targets or existing targets, and for the purpose of heart disease. And instead of relying completely on animal models, sheep, dogs, mice, etc., we are using human iPSC-derived cardiomyocytes as disease models. We use different ways to genetically alter these cells, to mature them into both 2D and 3D format. And then we use high-resolution imaging algorithms, machine learning algorithms for target ID. And then once we find a target we like, then we can also use the same platform for discovery of novel therapeutics. And importantly, this can be done in a modality-agnostic way. So whereas a lot of what we do on the left here of this slide is gene therapy and viral vectors, over here we can find both gene therapy targets as well as small molecule and biological targets with the precision medicine platform. So together we have 3 discovery engines, each representing different IP, different know-how, different capabilities. Our pipeline is organized along the lines of the 3 platforms. And what will strike you about this diverse and deep pipeline for a company our stage will call attention to a few things. One is that we have active programs coming out of each of the 3 platforms. 2, that the lead program is an IND-enabling studies, and that is for an AAV gene therapy for hypertrophic cardiomyopathy due to the MYBPC3 mutation. But you'll also see that we have gene therapy, different flavors of gene therapy, but also small molecules mentioned at the bottom. And I'll call your attention to the indication column and the size of population column. Some of the programs in our portfolio today are for genetic forms of heart disease, whether genetic dilated cardiomyopathy, genetic hypertrophic cardiomyopathy, genetic arrhythmogenic cardiomyopathy, and that affects thousands, tens of thousands, maybe even hundreds of thousands of patients in the G7. But you'll also see in our pipeline today indications like heart failure with reduced ejection fraction or HFrEF, heart failure with preserved ejection fraction or HFpEF, and chronic post-MI heart failure. These are very prevalent indications. These affect millions of patients in the G7. So quite a lot going on and under one roof here at Tenaya, multiple modalities, orphan and prevalent, and we have the opportunity to have multiple programs in the clinic in 2022. So how do we have these 3 platforms and how do we support such a diverse and deep pipeline of product candidates? And it comes down to capabilities. And what Tenaya, the decision we made early on at Tenaya is to internalize and to fully integrate multiple capabilities and approaches under one roof. And that includes disease models, capsid engineering, promoters and regulatory elements, drug delivery, and manufacturing. We don't have time to cover it all today, but in each of these, we have a focus on the heart that we believe gives us unique insight into heart biology and how these things intersect with each other. So with disease models, we have the ability to create novel models where they don't exist to mimic human disease for our target and drug discovery. Capsid engineering, we have robust efforts that have now spanned the last 2 years where we've done discovery, filed IP on novel capsids that outperform parental capsids for both cardiomyocytes and cardiac fibroblasts. We're doing our discovery in non-human primates as well as in silico machine learning-based design. Promoter and regulatory elements, similarly, we have been filed IP on multiple constructs that help us achieve more precise expression of genes in the relevant cells of the heart. Drug delivery, so certainly we will look at IV where that's appropriate, but we are not shy of exploring different routes of administrations, and we've even developed our own novel catheter for injection-based therapeutics, which is also in our portfolio today. Is manufacturing, and we'll talk a little bit more about it. But with the integration of these capabilities, we have the ability to deliver sorry, is the screen not moving? Yes. So we have the ability to develop products more rapidly. Sorry, we had a technical issue there for a second. So rapid prototype iterations as a result of the disease models, the capsid engineering, and the promoters and regulatory elements. As a result of the capsid engineering, promoters, and drug delivery, we can get to more precise product delivery, resulting in better efficacy and safety profiles. And then finally, with
drug delivery closer to the heart, as well as our manufacturing capabilities, we believe that will give us cost and quality at scale. So overall, the integration and internalization of these capabilities really supports our drug development in very meaningful ways. With that, we're going to turn the attention to manufacturing, and I'll present a slide and then turn it over to Kay. So why did Tenaya invest in AAV manufacturing capabilities? And there were three main reasons, I think, that drove our decisions. We'll get into more of that during the Q&A. But one is that we were pursuing orphan indications with high unmet need, where there's premature morbidity and mortality. And we know from the experience of other companies that there is an opportunity that if you have compelling data to accelerate rapidly through clinical development and towards a potential early launch. And we wanted to be ready for that even before we got into first-in-human studies. So that was one reason why we internalized manufacturing. Another reason is that, as we mentioned, we are actually also pursuing products that are intended for prevalent indications. And so for that, we know that the volume of drug that's going to be necessary is much higher than for orphan disorders. That means we need to be scale and to consistently produce products at larger scale. And it also means that we need to get to cost of goods that are lower than what is being done today for orphan drugs, that is appropriate for orphan drugs, and to get to COGS that can support commercially viable prices. And then finally, as you heard speaking about earlier, we're pursuing novel vectors, novel promoters, and regulatory elements. These can affect the properties of capsids. And we wanted to make sure that we had the manufacturing capabilities to support the transition of these novel vectors into the clinic. And so we could do head-to-head comparability on different manufacturing platforms and make sure that we understood the performance of our vectors very well and not rely on CDMOs for that. So for these reasons, we have internalized our manufacturing capabilities. And now I'm going to turn it over to Kay to begin to lay out some of what that means in our hands.
Thanks, Faraz. So when we started a couple of years ago, when we started really thinking about how we can plan out the manufacturing system, at that time, we wanted to pay extra attention first to the small scale the vector manufacturing, because those research grade, the vectors are the critical. Therefore, we wanted to come up with something to support the research and non-clinical development teams with the consistent quality, with the speed, including the stability and sufficient the quality control hold for the future further studies. A lot of cases, the people, running the research, and then later something comes up, and then we want to do a repeat. And then there is no more vectors available. Therefore, it is very important to have that kind of, take into account that the research grade manufacturing system. So we established the vector core, and then the vector core started working with the dual AAV manufacturing platform, which is HEK 293, as well as the SF9 for small and large animal studies. So we, at the moment, right now, we can provide the entire research and the non-clinical study. It doesn't really matter, the small animal or large animal, we can make that vector with the consistency and the flexibility. So once we finish the 50 liter, and then we start working on the 200 liter. So at the moment, we have the pilot plant operation has been established that can run up to the multiple 200 liter scales. And then if needed, we have a space to expansion. And then this 200 liter will cover for the manufacturing of all IND enabling tox drug product launch, as well as all size of the animal studies, including the non-human primate and then other large animal like the pigs. So we also at the start vetting the ideas to have the internalized GMP facility. So we recently, early this year, we signed up for the lease for the total 94,000 square feet warehouse facility, and then we selected to go with the modular GMP system. So we have a plan to be online in the second half of the 21st, and then to start the GMP campaign early next year. And we are target to provide all the details for the clinical development up to the initial commercial operation, which including the BLA. And for our internal, the GMP capacity, we have a two stage of the plant. The stage one, where we have a total 18,000 square feet out of the 45,000 square feet of the space to have up to two 1,000 liter scale. And then it can be, and then the stage two, we will planning to out to expansion, the rest of the 50,000 square feet, which we hope to have up to 10,000 liter scale, that we can have at least a couple of them. So we also secured the fully automated fill finish system within our facility under the same roof. So we have a maximum 2,000 vials per hour fill finish capacity is going to be established. So all of this, so we also have a time to evaluate all the many different kinds of the starting material, the cell lines and the bacteriovirus technologies. And then we have completed the licensing for the foundational IP to support all those things. That, and we, especially when we select the cell lines for the SF9, we had a plan in larger than 5,000 liter scale at the GMP scale. So before we getting into, yes, please go to the next slide. Thank you. So before we get into the serious development of the process with SF9, we wanted to test, is SF9 and bacteriovirus platform able to produce as good as the vectors from the HEK 293 in terms of the quality and the potency. So especially one of the AAV9 case, there was a very rare cases that has reported the AAV9 can be compatible. So we tested it in-house, and then at the preliminary testing, as showed, as you can see, the far left-hand side of the data, they did not compatible. However, we start working and then developing the downstream purification process to see if we can fix it. So somehow we came up with the proprietary purification schemes to preserve this, the infectivity as well as the transduction. So in the middle, you can see the both platform produced vectors produced very similar quality and transduction capability. So based on that, we further get into the in vivo efficacy study using the two different products, and then came up with a very similar in vivo efficacy as well as the quality. So these data gave us the confidence to move on with SF9 Vecuro as our major GMP manufacturing system. So next slide, please. So next slide. This slide showed how we are planning to scaling up. So for last several decades, the bacteriovirus can be easily amplified up to the 1,000 times. However, after you go after the several, the amplification process, the genetic instability of the bacteriovirus start losing your genome interest. So that could be a huge roadblock in the scaling up. So we have developed our proprietary, now it's patented, the bacteriovirus amplification schema that allow us to significantly scaling up without or minimum loss of our genome interest up to theoretically 25,000 liters. So we have validated so far from the flask, shake flask, to the 200 liter. But once we have a 1,000 liter scale and beyond, we will continue to validate our concept. So this actually allows us to unlimited scale independent production of the AAV in terms of the quality, potency, and the yield. So this is the very important discovery that we are very proud of. Let's go next slide. So next slide is showing what kind of the modular facility we have for our GMP facility. So this the modular part facilities can significantly accelerate this, the construction time. So within from the order to within 12 to 18 months, you will be able to get this modular delivered to your site and start running the GMP campaigns. So this has been validated by the other companies to pass through the pre-approval inspection and other major regulatory bodies. So this modular approach can not only save you time, but also this can give you a lot more flexibility in terms of the height, and then the capacity wise, we can easily expand it to the additional drug substance or drug product if needed. So this gives us the very high level of flexibility for responding to the market demand.
So I think that was the extent to which we wanted to provide Gbola company overview. Hopefully that gives people a sense of our vision, our strategy, our three platforms, the numerous capabilities we're integrating and internalizing, and then with a specific little bit of a dive there on manufacturing and early days for us compared to some of the folks where I think earlier than some of the folks who we have a lot of respect for at this conference we're presenting, frankly, many of whom who we learn from. We're also proud of the early commitment and investment that we've made as a young company in this area that is not following a fad, but it was necessary for the nature of the products, the nature of the innovation we're advancing, the nature of the pipeline we're advancing. We decided to make that choice to double down and invest in manufacturing capabilities. And so we could go on and on. We love what we do and could passionately go on about Tenaya, but I think it's a good point to pause and engage in some Q&A with you and with the audience if they have any.
Great. Yeah, thank you both. Thank you, Faraz. Thank you, Kee-Hong. It was really refreshing to see a comparison of HEC-293 with SF9 baculovirus. So well done on that. For your overall manufacturing platform, it sounds like one of the key differentiators is the end-to-end approach. What would you say as well are maybe differentiators versus what you see, or maybe what you're in terms of what you're aspiring to or where you are today? So what's different and what are the advantages?
I'll take a first crack at that, at least as it pertains to the whole company. I think the focus on the heart, right? That this is not something we're doing as one out of many things in our portfolio. This is all we do. And with that focus comes, I think, deep insight, including biological insight about the heart that leads to different ideas with regard to the different capabilities that we talked about, the capsid engineering, the disease models, the promoters, and the drug delivery, and even then how that translates to manufacturing strategy. So I think that's just something that sets us apart. And it's not just an aspiration. We, as I made a point to mention, we actually have filed IP on some of this novelty, or in some cases on the manufacturing side. We did file IP on some things, but other things we're keeping it as know-how internally. So I think it's the integration of the numerous capabilities is one thing. I'd also say that the vision here of modality agnostic drug discovery, again, focused on the heart and having the multiple platforms that are interrelated, but each provide their own unique shots on goal. So represent different opportunity sets, different risk events. I think the combination of all these things make us somewhat unique for a company our stage to our knowledge versus what others are doing today. So and then as you said, HEK SF9, that's not something, I've been in the gene therapy field now for more than a decade. Not everybody chooses to consciously compare head to head HEK and SF9 on every single product that we develop and every single novel capsid that we think that we're going to nominate as a capsid to potentially go into the clinic. I think that speaks to the level of, rigor, specificity, and not leaving things to chance, not assuming anything. That is a hallmark of kind of drug discovery at Tenaya. But let me turn it over to Kay to add to that. Anything else that you want to say, Kay, about what makes us different, unique, and refreshing?
Yeah, so I think that's a great question. So a lot of the company, I've been working with more than half dozen IND, and almost half of them, and plus one BLA that I worked with before, using the HEK with the triple transfection, dual transfection, and the producer cell lines and this and that. So I'm not against any of that. So, but the question is, how can we compare and select which one could be the best to fit for the purpose? For example, if we, as Faraz layout, we are not only targeting to the orphan indication, but also the prevailing indication. If we go to the prevailing indication, the cost of goods sold and the manufacturing cost are one of the significant portions. So the traditionally SF9 and retrovirus system has been studied for more than 3 decades. And then it has been studied a lot, but it's relatively new to the gene therapy. However, the technology and industry has been there for the production of the recombinant proteins or antibodies before. So what I can tell is that the biggest difference is the scalability and the yield. Because if you want to go to the prevalent, then you want to have to demand to meet like more than 100,000 doses. So there is a very difficult that we have known that HEK in suspension or adherent bioreactors and every systems can be very, very difficult to meet all of those high level of the demand. So that's why the SF9 retrovirus can kick in, that because it has been already proved that the scalability up to the 5,000 of the SF9, this insect cell line with the baculovirus since 10 years ago. And based on my previous experience that we have been working with SF9 in the baculovirus, it depends on how you design the process. So that's why at one of my slides that I talked about is the taking care of the genetics instability of the baculovirus system during the baculovirus amplification. That is super important. So based on that, we are very confident now. So we are very easily scaling up to greater than the 5,000, 10,000, 20, 25,000, whatever the market demand requires. So that is the one thing. And another one is there is a lot of concerns traditionally and historically that the vectors out of the SF9 is the less potent or less effective than the HEK. That is partially true. So I have personally tested more than 3 or 4 different serotypes I've seen. There are a few serotypes were exactly like that. It was not compatible. So it is very important to testing the compatibility before you select your platform. So how do we do it? That is very important. So that's why we started early to get the process development done for SEK suspension, adherent, and SF9 in suspension to provide a very fair purification, formulation, everything in the same starting point. And then we're figuring out, the different ways of the process development. So we were able to testing the compatibility out of the 2 different products with the 2 different, serotype, including 1 modified novel capsid. So we are pretty confident that if you can do that, the fair comparison, you will be able to get the clear distinction in terms of the quality, potency, and the yield. So that's what we have done so far, and we will continue.
That's great.
Yeah. So your presentation showed that your CGMP facility will be online in 2021. So can you talk a bit more about why it was important to have a CGMP facility online that early in your company's evolution?
Thank you. Yeah. So again, I'll take the first cut at it, Gbola, and then I'll turn it over to Kay to add. Look, everybody at this conference knows certain things to be true, just like as we do. So, at risk of saying the obvious, with the some of the things that went into our decision making were the same that I think any company has to confront. That the demand is outstripping supply. Supply is coming online, but it's uneven. And with the explosion of the space, the CDMOs are still playing catch up with supply. And so we wanted to have a little bit more. We all know that when demand outstrips supply, there's uncertainties related to cost, to quality, and to timing, or your ability to book a slot. And so from that vantage point, we, like many others, concluded that it would be good for us to have more visibility and more control over cost and quality and time versus 100% reliance on CDMOs. With no disrespect to CDMOs, because they play such an important function and we intend to work with them as well. But we wanted to have a little bit more control there. And then the other factors, Gbola, that we mentioned, the things that are a little bit more unique to us, the desire to scale to larger indications, the advancements of novelty, both in capsids and others. Again, that's something we didn't want to leave to chance, that leaving it for the CDMOs or external parties to have to work through the kinks of upstream and downstream process development, analytical development. So we wanted to internalize those things. So going to GMP was just the next step up in that. We had already done it for 50 liter and 200 liter non-GMP. Going up to the 1,000 liter scale as an initial starting point for manufacturing and establishing a facility was just the continuation of that thought process. And why we wanted to do it now at this stage is that we wanted to maintain maximal continuity between the process that we locked down for our preclinical development and IND enabling studies to first in human studies, all the way even into pivotal studies. And we thought we would have a better shot at doing that by making that change by investment now and producing the material for our first in human studies from our own facility with our own process, versus having to bridge later from, say, a CDMO into our own facility, maybe during pivotal studies at commercial. So given the news flow that we are all seeing, with the regulators really pushing back on changes and optimizations and things between phases, we thought that if we had a little bit more control and made these investments now, then what we took from our PD lab into the GMP facility and took into the clinic had a good shot of having some continuity going all the way out to an eventual, hopefully, a BLA, which we'll look forward to, knock on wood, in the future. So I think those are some of the factors that went into our decision to make the investment now. We had the support of our board and our investors to make this choice. In fact, it was very well received by investors during our last round of financing.
Sure. And speaking about all the investor interest you've seen recently, that's presumably coming at a time when a lot of smart KOLs are saying that heart is the next area for cell and gene therapy to emerge in a big way. But would you discuss how you'll handle the manufacturing complexities related to 3 different platforms? Obviously, you have a lot of opportunities doing gene therapy, cellular regeneration, and precision medicine. But how do you handle all that complexity as well?
Yeah, let me turn it over to Kay to respond to that.
Yeah, that's a great point. And, a lot of people get scared. However, in fact, we have strategically decided to use the outsourcing for the precision medicine, because there are very highly qualified CDMOs are available and CROs available. So we are outsourcing our precision medicine, small molecule manufacturing. But we do have internally expert in the medchem and the pchem, process chemistry. So we should be able to provide very good technical support for the external CDMO activities. So and then the regenerative platform and our gene therapy platform, those are actually using the viral vector system. So that's why we internalize the entire capacity from the vector core to the cGMP capacity internalized in-house. So we have 100% control and the preserving all those proprietary process manufacturing manufacturing science and technologies under our control. However, there is the key that we have to also understand that we do maintain very good relationship with the two leading CDMOs in the US, because what we see is during the clinical development, the speed and quality and consistency is a super duper important key to get to the BLA without any changes. Therefore, we will be able to ensure that clinical development up to the BLA or MAA, we can get to the consistency and the same quality with the speed. However, once we get to that BLA, then we cannot 100% produce all the commercial demand by ourselves. So that's the point that we should be able to exercise our outsourcing networks so we can transfer our validated process and assays and technologies to the CDMO, so the CDMO can focus on the commercial operation, just like what current the big pharma in the biologics world are going around. So that's why we keep the internal and external capacity together as our entire manufacturing network.
Got it. And then one of the big controversies or questions that arguably has been solved pretty well in recent years in AAV is how to deliver. So there's this dynamic of to what extent does your manufacturing influence the choice of local versus systemic delivery to the heart, or do you choose delivery first and then do your manufacturing scale up to support that choice? So please discuss that dynamic and how you feel it going forward.
So, Gbola, I'd say this is where that integration of the capabilities is so critical, right? Because I would say, and Kay and I agree on this, that we start with the science and we start with what's good for patients. And so with the combination of better vectors, with the combination of better promoters, we think we can get to higher expression per dose, let's say, with a and more exquisite control of that expression. With delivery, we can also get to, this goes back to that picture I was painting earlier on the capability slide, with the combination with the drug delivery, by infusing closer to the heart, we can hopefully get to, and this is in the public domain, lots of examples, people have done work in large animal models that you can get to lower doses and achieve the same therapeutic effect. So that's improving our safety margin. If we do those things, right, that we can get to higher efficacy, better safety at lower doses, then that helps manufacturing, right? And that certainly helps manufacturing, helps us maintain cost of goods, helps and reduces the load. But there may be indeed some situations where we're able to pull that off, novel capsid, promoter, delivery, and that works. Other cases, it may be IV. Some cases we'll be working with children, some cases we'll be working with adults. And so I think ultimately we let the science speak first and what's good for patients, and then manufacturing is the solve around that, and versus the other way around. We don't let the manufacturing constraint dictate what's the right science to pursue for patients.
Got it. And I think Faraz, earlier you mentioned something about learning from other companies that came before. So you're in a position of maybe being first for what you're doing, but having the opportunity to learn from others. So what have you learned? What are some of the growing pains that others had that maybe will inform what you're doing?
Yeah, I mean, look, first, we always stand on the shoulders of giants. So, and I think, people have tried gene therapy for the heart before. One very well publicized example is Celadon. And indeed, by their own telling, including in their published papers following the unsuccessful trials, Cupid 2 trials, manufacturing was considered one of the factors that, they had different processes from their phase 2A to phase 2B. They had different empty to full capsid ratios. And then there was always this question mark of how did that influence the product performance? So that's one example just within our field of how a changing of a manufacturing process from one phase to another can result in a product that looks different. And that, that was, information that we had back in 2015 and 16, and then flash forward to where we are today. The regulatory agencies are indeed calling out, don't want too many changes between phases of development. So that's something that certainly influenced our strategy. Certainly we learned from others. I can say, just having been in the field, worked at leading companies like Regenix and Bluebird, saw manufacturing challenges, saw, manufacturing runs not go well, saw challenges with CDMOs sometimes. I think that's part of the scar tissue that you accumulate by being in the field long enough. And Kay has a lot more of it than I do. So I don't want to call out any specifics. Celadon just happens to be an example in our space, but a lot of the folks over here saw and experienced challenges. Those have been shared in forums like the kind that you, Gbola, and the Chardon team have hosted here. And so we've absorbed those learnings and said, how do we do this better? I think we've also seen difficulties transitioning from academic, preclinical work done at an academic center, and then, great results that support your IND, but then your manufacturing's got to play catch up, right? And so I think we just decided to try to get as much right as possible up front, learning from everything that everybody else in this call and not on this call have gone through before. And we'll get some things right. We'll get some things, we'll make our own mistakes that hopefully we'll have the chance to share down in the future, but hopefully we'll get more things right than wrong because of the great work that's been done by others and that we've had the benefit to learn from.
Great. And then final question, what do you think is most underappreciated about the Tenaya story, whether it's manufacturing related or not?
I think the people are still don't fully understand just the tremendous research engine that we've built here with the 3 product platforms and what that really means in practice in terms of the opportunity sets and how much we can do, how deep the work is going on beneath the surface of our pipeline, plus the internalization of capabilities, and how much leverage and how much benefit we get by the intense focus on the heart. And I could give example after example, but I don't think until you've been on the inside and certainly under CDA, kind of looked at it on a program by program basis to really understand this capsid, this promoter, this delivery, and this manufacturing platform. And in one party's hand, if they kind of just took an off-the-shelf capsid promoter delivery and outsourced, what their product might look like versus what we know with the integration of all of these capabilities on one roof, how dramatically different you can have. The differentiation on a per product basis is remarkable, and how much insight we have because of the focus on the heart. I think it's one of those things that unless you've lived through it, unless you understand the complexities of the heart is complex, right? Complex physiological connections, complex mechanical movement. It's actively trying to push things out versus an organ like the liver, which is actually actively trying to absorb. So I think people don't fully understand that complexity. They don't fully understand the benefits of the internalization and integration of our capabilities and the productivity of the 3 platforms, but that's okay. I think people will learn more about it with time. But that's, I'd say, some of the things that people underappreciate about the Tenaya story. It's a positive underappreciation of how far we've come in a short time as a result of our capabilities and our focus and our strategy.
Great. Thank you, Faraz, and thank you, Kay.
Thank you, guys. Really enjoyed it.
We now conclude this session.